Ever wondered how cells duplicate their genetic material? DNA replication is the answer, but did you know there are two types, prokaryotic and eukaryotic? DNA replication is a fundamental biological process, the key to the propagation of life itself. At its core, it's about the precise duplication of the DNA in a cell's genome. This duplication allows for the transfer of genetic information from one generation to the next, enabling life to continue and evolve. In this journey, we'll explore the two types of DNA replication, prokaryotic and eukaryotic. Prokaryotic replication, occurring in bacteria and other simple organisms, is a relatively straightforward process. On the other hand, eukaryotic replication, found in animals, plants, and fungi, is a bit more complex due to the larger size and linear structure of the DNA. So buckle up as we delve into the fascinating world of prokaryotic and eukaryotic DNA replication. Prokaryotic DNA replication is a swift and efficient process that occurs within the cytoplasm of the cell. This process is a marvel of nature's design, perfectly orchestrated to ensure the survival of prokaryotic organisms. The journey begins with a single origin of replication per DNA molecule, a distinct characteristic of prokaryotic DNA replication. This singular point is where the DNA helix starts to unwind, opening up a replication fork. This fork is where the magic happens, the birthplace of new DNA strands. The process is facilitated by proteins, specifically DNA A and DNA B. These two act like the maestros of an orchestra, guiding and coordinating the whole process. They ensure that replication occurs smoothly and accurately, preserving the genetic information of the cell. In terms of speed, prokaryotic DNA replication is like a sprinter, fast and efficient. It adds around 2,000 nucleotides per second, a feat that speaks volumes about its efficiency. This rapid pace is essential for prokaryotes, allowing them to quickly multiply and adapt to their environment. Now, let's talk about the structure of the DNA. It's circular and double-stranded, a shape that is unique to prokaryotes. This circular structure is advantageous as it allows for a continuous, unbroken replication process. Unlike in eukaryotes, there's no need for DNA gyrase or telomerase because there are no ends to the DNA that need to be replicated. This simplifies the process, making it more streamlined. As the replication progresses, large Okazaki fragments form on the lagging strand. These fragments, around 1,000 to 2,000 nucleotides in length, are later joined together to form a continuous new DNA strand. The enzymes involved are DNA polymerase I and III, which play a pivotal role in synthesizing and proofreading the new DNA strands. Prokaryotic DNA replication, a quick and efficient process, crucial for the survival and multiplication of these simple organisms. It is a testament to nature's ingenuity, a process so complex yet so beautifully executed, ensuring the continuity of life on a microscopic scale. On the other hand, eukaryotic DNA replication is a more complex process taking place in the cell's nucleus. Delving deep into the cellular machinery, we find a meticulous process that ensures the accurate duplication of our extensive genetic material. Unlike prokaryotic DNA replication, which has a single origin of replication, eukaryotic DNA replication involves multiple sites for the origin of replication per DNA molecule. This means that the replication process can start from several points, speeding up the entire process. A significant player in this process is a group of multi-subunit proteins known as the origin recognition complex. These proteins identify the starting points for replication and initiate the process. Once initiated, multiple replication forks are formed in a DNA molecule. These forks are the areas where the double helix of the DNA is unwound, allowing the replication machinery to access the genetic material. The creation of the new DNA strands is coordinated by different DNA polymerases, namely DNA polymerases epsilon, alpha, and delta. These enzymes add nucleotides to the growing DNA strand one at a time, at a rate of around 100 nucleotides per second. This pace, though slower than that of prokaryotes, ensures the accuracy of the replication. Let's not forget about the Okazaki fragments. These short DNA sequences, around 100 to 200 nucleotides in length, are synthesized discontinuously on the lagging strand during replication. They are later joined together to form a complete DNA strand. Unlike prokaryotic cells, eukaryotic cells have linear DNA. This structure presents a unique challenge during replication. The end of the DNA molecule, known as the telomere, could be left unreplicated. To solve this problem, eukaryotic cells have an enzyme called telomerase, also known as DNA gyrase. This enzyme extends the ends of the DNA, ensuring the entire molecule is replicated. Despite being slower and more complex, eukaryotic DNA replication ensures the accurate duplication of our extensive genetic material. While both processes achieve the same goal, they differ in many ways. Both prokaryotic and eukaryotic DNA replication initiate with the unwinding of DNA, orchestrated by the DNA polymerase enzyme. Each process employs the semi-conservative replication pattern and begins with a short RNA primer. However, the location, speed, and complexity of these processes vary. Eukaryotic DNA replication occurs in the nucleus with multiple origins of replication. It's a slower, more intricate process involving numerous proteins and enzymes. The DNA strands are linear and longer. Okazaki fragments are formed. On the other hand, prokaryotic DNA replication takes place in the cytoplasm with a single origin of replication. It's faster and simpler, with shorter Okazaki fragments. The DNA is circular, eliminating the need for telomeres. So, whether it's the speedy prokaryotic process or the meticulous eukaryotic one, DNA replication is a marvel of nature. This journey into the world of DNA replication surely gives us a new appreciation for the complexity of life. We've delved into the intricate processes of prokaryotic and eukaryotic DNA replication, discovering the similarities and differences that help shape life as we know it. Both share an unwinding mechanism, utilize the DNA polymerase enzyme, follow a semi-conservative replication pattern, and initiate replication with an RNA primer. Yet, they diverge in the number of replication origins, the speed of the process, the size of Okazaki fragments, and the very structure of the DNA itself. It's these nuances that make the study of DNA replication so fascinating. For researchers in various fields, understanding these processes is paramount. It's the foundation for advancements in genetic engineering, disease treatment, and even the study of life's origins. Remember, every cell in your body started with a single DNA replication. Isn't that something to marvel at?